The bacteria and archaea have very simple genomes when compared to the eukaryotes. And also, when we compare them to the viruses, they have a much simpler basic makeup. All prokaryotic and eukaryotic, but all prokaryotic genomes are made up of double-stranded DNA. So this is probably the most likely candidate for the ancestral genome type. What did the first living organism's genome look like? It's probably something similar to modern-day prokaryotes. Almost all of them have a circular single DNA piece. That's their entire genome. Now, they can have, we can think of them as bonus DNA. They're not necessary for survival, but sometimes convey extra features, extra additional things that can be beneficial. And these are called plasmids. And they are also circular. They can be fairly large, but not as large as the original genome. And they can carry additional, like antibiotic resistance or other features can be carried. And in the lab, we take advantage of this using these to transform bacteria and add genetic material to laboratory organisms. Operons are a cassette, if you will, of genes. And so these are present in prokaryotic genomes. And basically what that means is there's a single promoter that will turn on 2 or 3 or 4 gene products. And usually all of those are involved in like a metabolic pathway. They're all very connected in that way. And so if you need one, you're going to need the others. And so they're all transcribed as one. And that's an operon. In eukaryotes, each gene is controlled individually. And then they have much more coding DNA as a proportion of their genome than the eukaryotes do. So roughly 85 to 95% of their genome is DNA coding. And so they have very little non-coding DNA. So that's the reverse of the eukaryotes. And although they are not common, there are a few types of bacteria that do have introns and transposable elements. In Unit 2, we mentioned 2 additional features that enlarge the genome of eukaryotes. And so those actually evolved before eukaryotes came around, but eukaryotes kind of magnified those, and they became a very important feature of eukaryotic genomes. So those introns and transposable elements are the 2 major things that have greatly enlarged the non-coding DNA of eukaryotes. Now, in addition, eukaryotes have this very common process called horizontal gene transfer. Sorry, I don't know. Prokaryotes have it. Eukaryotes do horizontal gene transfer, but it's much more rare. Where in prokaryotes, it's very common. And horizontal gene transfer can occur in a number of different ways. We use transformation, an artificial version of transformation, which happens in nature also in the laboratory. But there are active ways like transduction and then, like conjugation, and transduction, which happens through the action of a virus. Transformation is really more of a passive one where bacteria just takes up a piece of DNA and it gets incorporated into its genome. But all of these means that it's fairly easy, relatively speaking, to transfer DNA from one individual bacterium to another one, and sometimes even between very distantly related species. So one of the outcomes of this is that it's very difficult, in fact not even very meaningful, to do a phylogeny of the prokaryotes. And this is because any one individual prokaryote are going to have chunks of its genome that can be traced down different lineages. And remember that network-like or web-like pattern of relationships that we see among prokaryotic species is a big stumbling block for doing phylogenetics. And so that really means that we don't have a tree-like pattern for bacteria and archaea relationships. It's really more like a web-like and network-like pattern, and that requires different approaches than what we talked about in our phylogenetic section. And so as people began to get more and more prokaryotic genomes, they began to compare them together. And one of the first things they did, other than size, was to look at gene arrangement. Are the genes in the same order? And the short answer is no. Even between very closely related strains of bacteria, so we have 2 strains of E. coli here, there are some major differences. So every dot on this represents a gene. If it's a green dot, it means it's in basically the same relative order. If it's a red dot, it means it's been moved around. And notice there's an entire region here that's been flipped in orientation. Okay, if everything was identical, we'd just see a green line going from down here all the way to there, a little kitty corner line. And if we look at distantly related bacteria, even though they have maybe 90 or more % of the genes, they can be identified. We can find homologous, orthologous copies in both species. They are arranged in a totally different order. So every red dot represents a gene these two species have in common, but notice there is no clear pattern of synteny. Okay, so synteny is the order of genes in a genome. And so what this means is there's very, very frequent genome rearrangement among the bacteria and the archaea. And it's so common, in fact, that even though a bacteria might have 90 or 95% of the same exact genes, there's no discernible order in which those genes are found in the genome. And so genome rearrangement happens quite frequently. It usually doesn't have much of a phenotypic effect. Sometimes it's kept, sometimes it's just removed by chance. But enough of them are kept to completely rearrange genomes over pretty short spans of evolutionary time. Another thing that we found as we started to investigate these genomes is that there's often a trend of codon usage bias among prokaryotic genomes. And what this means is that if there are 4 codons, for instance, that some of them are used more frequently than chance would predict. So instead of 25% for all codons, we might see one that's used much more frequently. And we've talked about this and how this might affect our model of evolution, right? But we didn't really explain what caused it. So let's look at it. What people noticed is that these biases also tend to match the tRNA frequencies, the amount of different tRNAs that were found in the cells. tRNAs are transfer RNAs that bring in the correct amino acid for the specific codons. And wobble plays a little bit here. We won't get into that. If you know what wobble theory is, great. If not, don't worry about it. But this was the first clue that this matching tRNA frequency might be a clue to the reason why we had codon usage bias. And so the main idea is that it increases the speed and accuracy of protein translation. And think of it this way. If you have 4 different things you can grab from all to get the same end result, it might slow you down a little bit. There's a little hesitation. Do I grab from this box or this box to get the red Lego? And if there are red Legos in both of them, it really doesn't matter. But there's that slight different hesitation. And so the same thing, if we're bringing in tRNAs to build these proteins and there's mainly 1 available rather than 4 equally, it's going to speed it up a little bit. And so most likely when we see a codon usage bias, it's something that has increased the accuracy and replication of transcription. The other thing that we notice, the other trend, and we talked about this with models of evolution, is GC content bias. GC content bias has many, many explanations. We're just going to look at 2, but realize that there are others. These are 2 common ones, but there are many others. Actually, we'll just look at 1. But these can be categorized under different heads of the different titles for what category they fall into. And they're also cool because they can help us identify horizontal gene transfer events. And we'll talk about that here in just a little bit. But notice the range. Some bacteria have almost 75-76% GC content, where others have a much lower one. Plasmodium is a protozoan, not a bacteria, but other bacteria have very small ones. We don't have a bacteria species here with small ones. But the reason they help us identify horizontal gene transfer event is there are selection pressures, a wide range of them, that lead to these codon usage biases. And one of the very common ones, the only one that we're really going to talk about, is the fact that different GC content bias lead to different levels of stability of your DNA. So G's and C pairs have 3 hydrogen bonds, whereas the A and T pairs only have 2 hydrogen bonds. So that means if you have more G and C's, you just have more thermodynamically stable DNA. So if you're living in an environment, high acidity, high heat, something where stability is good, the natural selection will favor more G's and C's in your genome, and vice versa. Now, there are many other explanations for GC content bias. We're not going to look at them. But just realize, and so if we have natural selection causing a bacterium to have a certain GC level, say 80%, and then a gene from that bacteria gets transferred, or maybe a chunk of DNA gets transferred from that bacteria into a distantly related one with a much lower GC content, it's going to take a long time for natural selection to bring that new DNA in line with the GC content of the new host. However, so for that short period of time, if we see an area of very elevated GC content, we know that that's a horizontal gene transfer event. So that's kind of a useful feature of this factor of prokaryotic genomes. And basically what it tells us, and also trying to do phylogenetics, is that this is rampant across prokaryotes. And especially if we look at it over the billions of years of history, it's happened so much that it even obscures relationships amongst the different types of bacteria and archaea. All right, let's look at eukaryotic genomes. Now in eukaryotic genomes, we have some different physical architecture, different rearrangements. There are nuclear portions of the genome, which include all of the chromosomes. And then we have organellar genomes, and in all eukaryotes, there's at least one organelle. Animals, for instance, have the mitochondria, and that's the only organelle they have. Other organisms have more than one. Plants have the chloroplast with its own genome and the mitochondria with its own genome. And then there are other organisms that have more. There's some rare organisms that have 3 or 4 different organellar genome. So we need to look at both of those. The organellar genomes are usually only a very small fraction of the overall genome, though. And what we find as we look at it is most of the eukaryotic genome is non-coding. Oh, by the way, a little bit of an aside. Organellar genomes are most similar to bacterial genomes. And that should be a no-brainer for you, right? Where did the genome of the mitochondria come from? Well, the mitochondria and its genome were a bacteria that began a symbiotic relationship with an early eukaryote. Same thing with the chloroplast. So the reason the mitochondria genome looks more like a bacterial genome than the rest of the eukaryotic genome is because it originated as a bacterial genome. But among the rest of that genome, most of it is non-coding. In humans, you get 98.5%. And if you look at the numbers, about 25% of the genome is introns. About 1.5% is genes, and the remaining, most of the remaining, at least 75% or so, is an origin from transposons, duplicated DNA that's been put into the genome. Now, just like bacteria, eukaryotes have very, very frequent genome rearrangement. So even very closely related species, although they may share 90 or more percent of their genes in common, they can be missing some, they can have regions that are flipped around, they can have genes that have been moved to a very distant part of the genome. And as we look about cross-distantly more related species, it's even worse. So this is corn and rice, 2 monocot plants, and an attempt to line up or to look at the synteny of their genes. And notice there are regions that match up, but there are regions that have been shifted around greatly. So every line here, every little split represents a shift in portion of the genome. And so genome rearrangement is frequent across all organisms, not just the prokaryotes. It happens all the time in the eukaryotes also. Now, a spliceosome is a term used for this discrepancy between protein coding regions and protein products. So although we might only have 20,000 protein coding genes, we might have 30,000 protein products. And this is because of differential splicing. And so the spliceosome is what recognizes regions to splice, and because of differential splicing, eukaryotes, especially mammals and complex organisms, can have many, many different protein products from a single protein coding gene. Now, introns themselves play a role in the evolution. And we can see this because there's high levels of intron conservation. That means if I compare the introns of a gene in a human to maybe a chicken, I might see a similar arrangement, which means there's been selection pressure to maintain that arrangement of the introns. Introns also play a role in crossing over and recombination. They play a role in exon shuffling, and so they play a role in the diversity and the architecture of eukaryotic genomes.